I guess since everybody's sitting back here, I'm going to have to come to you to do this introduction. So, but I'm pleased to introduce Anadi Krishnan. I know the fellows and others have had a chance to work with Anadi, but perhaps some of you have not. Anadi came to us a couple of years ago when she and her husband moved here from Penn State, both having done their doctorates there. And Anadi had contacted me because of her background in biomaterials, engineering, and thrombosis, and also an interest in clinical cardiology. Had done some imaging research and some work with nitric oxide and a variety of things that made her a very attractive candidate, and I thought it would be a perfect fit for Duke and the DCRI. She then applied for one of the Bugher fellowships, which is offered through the American Stroke Association, and was awarded that fellowship, and in that context was working with Larry Goldstein in neurology and going on rounds and doing a variety of projects with the other Bugher fellowships. And in addition was working with us on a project in the Sullinger Laboratory on a more of a translational science background. We asked some fundamental questions about some of our aptamers and how they might work under varying conditions that could be applied to neurology, could be applied to coronary bypass surgery, and applied to a variety of settings that were not the traditional settings. So Anadi took that on and spearheaded it, and she's going to talk a little bit about that work today. So it's a pleasure to have her with us, and please ask any questions that you might have, and we're going to save plenty of time towards the end to get your comments and feedback. So, Anadi.
Thank you, Dr. Becker. The work I'm presenting today is actually a small, focused segment of a larger translational effort that Dr. Becker mentioned in studying a novel, regulatable anticoagulant system with potential application in stroke therapy. The basic premise of the work is from the challenge in striking that fine balance between preventing ischemia and causing bleeding. And this is particularly challenging in stroke given the varied clinical manifestations of stroke and the additional vagaries that come from antithrombotic treatment and hemorrhagic transformation that comes in stroke in particular. So here is a framework to just what guides an optimal antithrombotic choice in stroke. Here it's showing it's the specific site of pathogenesis. I guess I'll point here. The specific site of pathogenesis, which can vary from cardiogenic or arterial sources, the type of ischemic stroke, and here's showing atherosclerosis of the large and small vessels that supply the brain, being one of the major causes of ischemic stroke. Then there is cardiogenic emboli with atrial fibrillation and valve disease, and the near 30% of ischemic stroke being cryptogenic, despite a very thorough evaluation. And the third factor would be to identify specific groups of patients who are at increased risk of disease, increased risk of stroke in this case. So here, it's a combination of these three factors that guide the optimal antithrombotic choice. But it's still a difficult task, and here is an important chart from International Stroke Trial summarizing thromboembolic and hemorrhagic events. This is while evaluating heparin and as randomized between aspirin, heparin, both, or neither. And you can see that most favorable results are with low-dose heparin and a combination of low-dose heparin and aspirin, and you get high rates of really unacceptable rates of bleeding with high doses of heparin. So as a result, the anticoagulation is not considered first-line treatment in stroke, and it's not advisable as part of the guidelines. But what is advised, though, is thrombolytic therapy within a very brief window of about 3 to 4.5 hours. But what that does is limit the number of patients who can avail TPA, and it's only about 1 to 6% of stroke patients who become eligible for thrombolytic therapy. So then again, most patients are then treated subsequently with an antithrombotic agent, and as you can see here, with aspirin, antiplatelet therapy. But then that still leaves a fair number of patients who benefit from anticoagulant treatments, and in this case, the heparin-protamine pair. And the heparin-protamine pair has its own side effects that you all know of. And so that's what brings me to an ideal anticoagulant, one that Dr. Becker has talked time and again in conferences here, and you all are aware of the concepts behind. So I'll go into the background a little bit and then introduce my own results. An ideal anticoagulant, as the framework here, is a drug antidote paradigm, and is one that affords us the opportunity to specifically and actively control the degree of coagulation that we want in a system. And in this case, that is achieved through a unique set of molecules called aptamers. And aptamers are synthetically designed. They're oligonucleotides that bind specifically to any given protein. And in this case, a key coagulation protein, factor 9A. And by binding to factor 9A specifically, it inhibits downstream clot formation. This is ideal in its own right, but what makes it exceptionally novel is the concept of an antidote that it can turn off the drug response. And that's the antidote here is just simply another oligonucleotide sequence that binds in a complementary Watson-Crick base pairing fashion to the aptamer and completely turns off the drug response. So that's the unit, the aptamer and the antidote concept that drives regulatability of anticoagulation. Factor 9A here is an important target. As you can see, it's the key enzyme between multiple coagulation pathways, and it's also the key enzyme necessary to produce the critical quantity of thrombin necessary for clot formation. So it's a very important protein, and targeting factor 9A affords us the opportunity to control coagulation and upstream, like in one of my previous slides, it's upstream of where heparin would act. So it gives us a better opportunity. So aptamers are generated through a process, a series of steps here called the SELEX process. You begin with a group of library of RNA. You incubate them with the protein of interest. And again, this can be, in my case, as I'm mentioning here, it's factor 9A, but this can be any given protein that we want the function to be inhibited for. So here, this would be factor 9A. You incubate the nucleic acids and the factor 9A together. You separate out the ones that bind versus that do not, and then you standard amplification processes that go through giving you the sequence of the highest affinity for the protein of interest. And and you continue this process even post-translational modifications that will result in in the final aptamer sequence that you're interested in. It has better half-life. It can increases affinity. And then the two examples that I will go into in this work as side chains are two modifications are usually cholesterol and polyethylene glycol as side chains to the native aptamer, molecule. So here it is again. So I just went through quickly. So you have the factor 9A protein, the factor 9A protein, and here is the aptamer that binds to the factor 9A molecule, anticoagulating the system because it keeps the factor 9A away from binding to its target, and the antidote that reverses the anticoagulant effect. And this reversal has been shown in human studies to be within 1 to 2 minutes in a rapid, consistent fashion. So I should mention that much of this work, the early ideas have all been attributed to Dr. Bruce Sullinger and Dr. Chris Rasconi at Duke, the Medical Sciences Research Building, whose lab I have used and who have been extensive support for me throughout the work. And my own focus with aptamers was to study thrombosis, coagulation on non-biological surfaces, and a host of surfaces that you see here all have the potential to clot, aside from independent of the in vivo system of coagulation, and over a range of sophistication, as you can see here. So aptamers is an important context in non-biological surfaces. So and the way this happens is that the surface activates factor 12, one of the key coagulation proteins, and through a contact activation mechanism, and potentiates down the cascade through to form a clot downstream. So this is the intrinsic pathway of coagulation, and that's the focus of my work for the rest of the talk. So here is a setup, and I'll go through this in detail. The specific objectives here for me was to understand aptamers in the context of surfaces, as I said, so with varying surface chemistry, surface energy, and over a range of aptamer concentration. So then, with that in mind, you would then focus on the intrinsic pathway. Then that would mean that the experimental system would be a plasma, and in this case, saturated plasma. And so you have the plasma, you have your surface activator, the activator of interest, and then you initiate clotting with what's called recalcification, or you introduce calcium chloride back into the system in a saturated system, and you initiate coagulation. And you measure clot time from the time of initiation of coagulation to the actual formation of clot. So it's a very simple system, but it's a very targeted system where you can define what you want, what goes into the system, and you measure the response that you want out of it. So basically the steps are that you initiate coagulation with the surface activator and your calcium chloride, and plasma, of course, is the system, and you wait until you see a gelation or the clot formation. And we, when we ventured on this study, as Dr. Becker mentioned, Dr. Becker had experience with cardiopulmonary bypass models, animal models, and there were certain hurdles with application of aptamers in the context of surfaces in a cardiopulmonary bypass system. So that being said, surface is one aspect. And the other important aspect that we thought in a CPB model would be temperature. So we took these two factors, surface and the varying temperature, as two factors that we wanted to study how aptamers would behave. Are they, is the aptamer still functional in a system where you generate coagulation through the contact activation mechanism, where surface is the only mediator. And so that's, here is a proof of principle curve. I think this one curve can actually talk about all the work, but here is showing clot time as a function of surface area. So all I'm doing here is varying the type, varying the area, the amount of surface that goes in. So that means you're varying the degree of activation. So low surface area would mean low activation. High surface area would mean high surface activation. So you can see there are two trends. One, that the clot time goes down. If you just look at the very bottom one, which says control, you see that the clot time goes down with increasing surface area, the clot time decreases. And the other trend that you want to look at is that when you add the aptamer to the system, you see a very graded increase in clot time with increasing concentrations of the aptamer. So two things, that surface activation is an important factor here. The degree of activation controls the degree of clot time, the degree of response you get. And that response can be further controlled by the introduction of the aptamer to the system. With increasing concentrations of the aptamer, you get increased delayed coagulation. And so here, but that might, it sounds very simplistic in some ways, but what we found remarkable was that when you varied the procoagulant, the surface, if you had a different material, what we found, the degree of the anticoagulant response we received was different, that you measure was different, and it was distinctly different in these two cases. The top panel here is showing glass, and it's just the most procoagulant surface you can have, and you're getting about fourfold from baseline. You're getting about fourfold increase in coagulation time. And here it's actually comparing two types of aptamers, the cholesterol and the pegylated aptamers that we spoke about earlier. And here the lower panel is polystyrene, or it's representative of any polymer film used in biomedical applications. And what you find is that you get about nearly 25-fold increase in clot time. So it's more than just the quantitative increase. What it's telling us is that the difference in activation from the surface controls the measured response of the aptamer. And so it's just, this graph is collecting both of those together, and this is showing the most activating surface, the surface that represents a high degree of activation. Here is a surface that represents a lower degree of activation. And the anticoagulant response here is much smaller than what you see with the polymer surface. So I'm calling this anticoagulation efficiency, and the efficiency of anticoagulation is lesser with a stronger activator versus that of a lower activator. So that was an important finding for us, that surface characteristics control the efficiency of anticoagulation. Some of it is anticipated, but then it was important for us to quantify the difference between the degree of activation. The next important thing, as I mentioned, is the temperature, such as in a CPB system. So we evaluated coagulation or anticoagulation in varying incubation temperatures, and this is just it's very simple, and it's just showing that anticoagulation efficiency actually falls off, decreases with decreasing incubation temperature, with decreasing temperature. And this is, again, it's a bit of you're extending this to a CPB system, but it's still important to know and to have a feel for that the effect of the aptamer might be potentially reduced in cardiopulmonary bypass or hypothermic conditions. So that's that. And these are just the two graphs are showing two different aptamers. And so that together, both of those observations together bring us to highlight that aptamer binding efficiency, how the aptamer binds to the factor 9A protein or any given protein, the complex formation is a very important factor that governs the anticoagulant response. So this is not my work. This is citing work from literature, but it's clearly showing that the complex formation is much more efficient than at a higher temperature, than at a more optimal temperature than at lower or hypothermic conditions. Almost nearing the end of my talk here. So these are the three factors that control anticoagulant response. Main feature is the intensity of activation. If you're studying an in vitro system or a system such as in a CPB model where you have both in vitro and in vivo activation, it's important to understand that the intensity of activation is a significant parameter that controls how much anticoagulant response is achieved. Aptamer concentration, of course, is an important factor, and the aptamer factor 9A binding efficiency. So this graph collects everything together. This is showing anticoagulation efficiency as a function of aptamer concentration and surface area, activator surface area, which is representative of the degree of activation. So you can see that the anticoagulation efficiency is highest at high aptamer concentrations, and it's in fact lowest at high degrees of surface activation. That brings me to wrap up. It was part of my, like Dr. Becker said, it was part of my Bugher fellowship. We wanted to translate the aptamer concepts into a stroke-related application, into something of a CPB model. So it was a translational study. It focused on the basics of stroke therapy. It actually helped me focus on antithrombotic guidelines with Renato here during my fellowship. So it helped bring all of that together, and it helped bring the aptamer into a stroke picture. And of course, the in vitro system demonstrates a function of this novel anticoagulant, and it's been evaluated on different surfaces, and different temperature conditions. So, any questions? I'm open to questions.
Done, Anandi, and the one thing that I can say is that I take no responsibility for any of these ideas. They were all Anandi's. The only thing that I did was ask a question about how an aptamer may behave in a different system, and the rest of it she took from there and applied a lot of the background that she had. But fundamentally, when we started to work with aptamers, and some of you in the room may be aware, going back almost a decade now, that one of the first indications for the aptamer antidote pair that was discussed was cardiopulmonary bypass, because of the fact that it's one of those environments where you have to anticoagulate, and you have to anticoagulate aggressively. But then at the end of the case, when you're getting ready to close and you take the person off of bypass, you have to reverse those effects. So the thought was an aptamer antidote pair would be perfect under those circumstances, and we had done some small or intermediate size animal models that suggested that indeed the aptamer could be used. And as we got to larger animal models, there were some questions that were being raised about whether one could use an aptamer. So Anadi has uncovered some very, very important translational information here, and just to summarize, that the surface is very, very important in terms of how effective the aptamer is. And for those of you that have been around cardiopulmonary bypass or extracorporeal membrane oxygenators, you know that there is a very, very broad surface of meters in length of varying materials that the blood and an anticoagulant has to course for the patient to remain anticoagulated during the case. And in cardiopulmonary bypass, we cool patients. It's deep hypothermia, so we're down into the the low to mid-20s. So it would seem, and this is something that needs to be tested further, that with the surface and the activators that are present in bypass and the deep hypothermia, that indeed it may change the dosing for aptamer use. It may change the overall kinetics of how the aptamer binds onto its target protein. It changes the folding dimensions and dynamics, which are very, very important. And there are certainly other clinical conditions where hypothermia is discussed. I mean, following cardiac arrest, hypothermia, although not to these degrees, in any type of extracorporeal system or circulation, hypothermia is more the rule than the exception. So this is very, very important information, something that the Sullinger team and others have paid attention to with great interest, as one might imagine. So, Anadi, what do you think are the next step in your research? I know that you're doing some additional experiments to try to solidify some of these observations, but what's your plan at this point?
With this work, I'm hoping that this is an important foundation for studying further on surfaces. You could alter the type of surface, you could introduce cell-based models and evaluate the contact as well as intrinsic and extrinsic mechanisms, and study factor 9A aptamer in that context. And moving forward, like you said, to a CPB animal model or just a extracorporeal circuit model would also be an important one, where you can alter the surface, alter the temperature, pursue this to a more next level of complexity.
Yeah.
Would be one direction.
Yeah. Unfortunately, as many of you know, there are a number of models on site at Duke, over in the vivarium, as well as in other parts of the campus, that there are enclosed circuits. There are animal models for cardiopulmonary bypass. There are animal models for left ventricular assist devices, and a lot of potential avenues that one could pursue based on this work. What about stroke? I mean, you started out by saying
right.
that part of the interest here was around stroke, and we would agree that anticoagulating patients with stroke has been difficult for a variety of reasons. Do you think that having the antidote aptamer pair gives you greater flexibility than you would have with any other anticoagulant?
I would definitely, yes. And I've been, as part of the fellowship, had a chance to present at the neurology stroke conferences, and it's time and again it comes up that it's a complex problem to be able to anticoagulate. And even though a significant number of patients could benefit from anticoagulation, because of the untoward side effects of heparin, anticoagulation itself has been set aside as an option. So it leaves a significant number of patients who could benefit from it. So the aptamer and the antidote, with the tight regulatability that it affords, will definitely be an excellent tool for stroke patients in acute care, at least for specific subgroups of patients who could benefit from the combination. It's definitely, given that it's you're always finding the balance between ischemia and hemorrhage, and ischemic stroke and hemorrhagic stroke.
Yeah.
So it's important tool for neurologists, I think.
Yes, I agree with you, and it's not solely for the patient who has a cardioembolic stroke, where we do tend to anticoagulate them, and then we always watch carefully for signs of hemorrhagic transformation, and then if there is, we wonder, what are we going to do now? Obviously, you can reverse heparin with protamine. There are some potential downsides of protamine. And work that's ongoing in the Sullinger laboratory with Annie Laskowitz and others that are looking at stroke models, one of the things that's being tested is that in a hemorrhagic stroke model, that if one were to utilize the aptamer under those circumstances, that you might be able to reduce the amount of damage that is incurred because of how rapidly you can reverse the anticoagulant effects. And so something that could be used there. As far as whether it could be used in non-cardioembolic stroke settings, I suspect that it could. One would probably have to approach that with some caution, even though it's a reversible anticoagulant. But what are your thoughts there, having presented it to the neurologist?
I think, definitely atrial fibrillation, that's the group that one would focus on mostly. There is, heparin is rarely used now, I think, in stroke conditions. And there are one subgroup that I had come across when we were doing the guidelines too is pregnant women or venous thromboembolism. Those are specific conditions that I'm thinking that the Aptamer antidote, again, given in that pair with the tight regulatability, would be useful. Mm-hmm. Yeah, but largely it's the cardiogenic emboli, atrial AFib group, and valve disease, prosthetic valve patients who could all benefit from the tight regulatability there, anticoagulation there. Yeah.
Let me just briefly explain to the group about the PEG versus the cholesterol. When we started doing the work with Aptamers, the one thing that we tried to achieve was a long circulating half-life, because the Aptamers that one could produce have half-lives that are seconds or minutes in duration. But the thought was that if you had a compound with a long half-life and you also had the benefit of the antidote, that you could develop molecules that had half-lives of days or weeks, and you wouldn't necessarily worry about it because you could always reverse the effects. So cholesterol was the first side chain that was tried, and Anandi, you showed some of those data. And then the second was PEG, or polyethylene glycol, which is a common side chain that helps the molecule stay in the circulation for a longer period of time. But the question that I have for you, Anandi, is do you think that in the setting of cardiopulmonary bypass that we'd be better off utilizing just the Aptamer itself without a PEG side chain, because that may be what's binding onto the surfaces and then changing the shape that prevents the binding onto the target, or to use cholesterol, since your data suggests that perhaps the cholesterol side chain creates less of a stoichiometric problem with the binding onto factor IX as your target?
Yeah, that really is an important question, an important observation that we got from our work. Yes, I would think so, but then we do have the disadvantage of the half-life problem. But then the cholesterol side chain Aptamer seems a lot more robust in my experiments. The PEG-related Aptamer as well, and it has its own advantages. But in a setting of, say, hypothermia, at decreased temperatures, the polyethylene glycol side chain is probably altering the Aptamer binding as well. Mm-hmm. And so I would definitely think the cholesterol side chain is a nominal choice.
So you may have to change your preparation according to the indication, where the PEGylated molecule works well for percutaneous coronary intervention or acute coronary syndromes, as is being tested right now in the RADAR trial. But in some of these extracorporeal settings, bypass in particular, or it could be for the purpose of dialysis or ECMO, that's used more often in the pediatric world than it is in the adult world, that you need a variation of a theme just because of the different properties. So I'll open it up and see if people have questions or comments. You probably do. Diana's becoming an expert in Aptamers over the years.
Do you find that there's any changes in the pharmacodynamics of the binding of the reversal agent to the Aptamer with temperature or surface, or if you're using PEG or cholesterol?
So, no, I actually haven't studied the antidote. I focused only on the Aptamer per se. Just given, to answer broadly, I would think that the sequence aptamer and the antidote are complementary. And so I would think that especially temperature might have some effect on the antidote, the sequence of the antidote or the folding of the antidote as well. But I haven't studied that in my own work. But I have definitely seen, like Dr. Becker pointed out, the polyethylene glycol side chain aptamer was the one that showed some reduction in its binding or in its anticoagulant effect. But it's not so much, it's not drastic to say that the effect is lost on us. So I'm sure there'll be a moderate change, but the change is probably not very substantial.
I think you've touched upon what some of the next experiments may need to be, but I agree with Anadi that the antidote is not pegylated. So if it's the peg that's causing the problem, we will probably be okay in terms of being able to reverse the effects, but that's yet to be shown in the next series of experiments. I think it's a good question. Other questions or comments? Yes, John.
Anadi, that was a very nice and interesting talk. Thank you. Two questions, both of which will probably display my ignorance about stroke and aptamer biology. But first question is, there's a fair bit of damage that's associated with ischemia that's inside the blood-brain barrier. There's also microvascular coagulation, other effects that happen in stroke. Does the aptamer cross the blood-brain barrier and have an effect in this ischemic zone outside the sort of direct vascular clotting cascade?
I'm going to attempt an answer before passing on to Dr. Becker. But so the aptamer is an oligonucleotide. It's a small chain of nucleotides. This one in particular is about 35 base pairs. So it's a small molecule. Even given the pegylation, it is a small molecule. So I would think that it would cross the blood-brain barrier. But then, so the target, the important thing here is it's a targeted molecule, and its function in blood or in circulation is to be bound to its target protein. And it has very high affinity to be bound to factor 9A or any of its target proteins. So I do not think that would happen, but then there is a chance.
I agree with your response, and my answer is yes and no, that the aptamer does, but the aptamer attached to a side chain like cholesterol or polyethylene glycol does not, just because of the size. I mean, you get up to about 15 kD or so, and it's just not going to cross. It's too big a molecule at that size. And but the smaller molecules might, but they are so specific in terms of their effects, unless there was some wayward factor 9 or something. It doesn't bind onto anything else that we're aware of. It doesn't bind onto thrombin or vascular proteins or other things that may be in that environment as you described, John.
Thanks. Second question was, and again, this is just out of my own ignorance and more actually curiosity. Does the aptamer get into the cell, and does it alter synthesis or translation or anything in the signal transduction pathways or receptor trafficking? Anything that happens in factor 9 biology?
Not in its the design, not in its current design as we spoke here. Again, it's the process is simple, focusing on the SELEX procedures. So you begin with that nucleotide sequence. You begin with a group that you know or you anticipate to have a high affinity for the protein of interest, and you funnel that down until you get the highest specificity or highest binding with the target molecule. So this is designed to be in circulation. It's designed to be bound to the coagulation protein, or in this case factor 9A, which is an activated factor 9 molecule. So do not anticipate that it would cross into the cell and actually initiate or involve with production of factor 9 inside the cell. I do not anticipate that.
Oh, right. And John, you're absolutely right. The initial generation of aptamers was designed as siRNAs. Mm-hmm. So they were carried into the cells, and they would change signal transduction, they would change gene expression, and they're still being used for those indications now as for oncology indications and other indications. But the work that is being done currently is solely at the protein level, the small molecule level. Is not you can attach it to vectors and other ways to get it into the cells, and that's still done.
So thank you. That's very interesting.
Thanks. Okay, now that everyone is an expert on aptamers and stroke and materials, I'd like to thank Anadi for the very innovative work and highly translatable work that she's done over the last couple of years. It's something I'm sure I've said before to fellows and others that do research. Anadi's presentation took about 20 minutes. It's 2 years of work at the bench and having weekly research discussions with myself and Bruce Sullinger, Jens Lorman, other people in the Sullinger laboratory. So it's a lot of work that goes into a brief presentation, but the translatability is something that certainly carries on. So thank you for all your hard work, and thanks to you for attending today.
Thank you, Dr. Becker. Thank you, everyone.